So, good morning everyone. My name is Michael Holtzhausen and I am a nursing student from the Davao Doctors College, and I will be showing you and explain the straight plate method lab and of course how I do it. So, straight plate method is a common method for the isolation of the pure colonies from a mixture of plates, isolating a pure stain from a single species of microorganisms, often bacteria. Then samples can then be taken from the resulting colonies, and a culture can be grown on a new plate so that the microorganisms can be studied, identified, or tested. Okay, so when performing this procedure, we must keep in mind that we want to dilute a small particle to a point where one portion of a petri dish has just one bacterium. The equipment that we need to use for the straight plate method are the sample containing of a mixture of bacteria, a petri dish containing solid, medium, or agar, the wire loop, and a Bunsen burner. So first, we must first cool for a couple of minutes the sterilized loop from the Bunsen burner. Then we must touch it to a sterile plate. Then dip the loop into the sample containing a mixture of bacteria to pick them up. We actually divided the area of the dish into 4, and that is where we strake the wire loop. Then we take from the sample containing a mixture of bacteria, then streak the loop in the first area. Then after that is to kill the remaining bacteria on the loop by the flame of the Bunsen burner, then let it cool again, and this time is to drag the loop through the previous path of the first quarter, then proceed to the second area. Okay, and after that is you just have to repeat the last procedure. Kill the remaining bacteria by flame, let it cool, then drag it in the third area. It is the same for the fourth area. It's the same procedure for the fourth area and the third area. Okay, so in each path I observed that the loop picks up a smaller number of bacteria, and it can spread farther and farther apart of each other, getting smaller and smaller and spread further apart. Like for the second path is smaller than the first path. The first path of the stroke. The third is smaller than the second path, and the fourth is smaller than the third. And then during incubation, the bacteria multiplies and produces colonies. Each consists of millions of cells derived from the single parent bacterium. Then the isolated colonies found in the last streak represented the isolated strains to ensure the strain's purity. Then the straight plate procedure is usually repeated a few more times, using an isolated colony as a source of starting bacteria. Okay, so now to further understand what I have just said is that on this picture, pure culture can be obtained by picking well-isolated colonies and restricting this on a fresh agar plate. Then the biochemical tests are used to identify bacteria. These tests are only valid when performed on pure cultures. So we sterilize the loop on the flame like this and then let it cool afterwards. Then we must pick for an isolated colony like this picture. And then spread it on the new agar plate on one of the areas or quadrants using on a zigzag motion po siya dapat. Then flame the loop again and then let it cool. The easiest way to make sure that the flame has cooled down enough is to gently touch it to the agar and make sure that the agar does not melt. Now turn the plate to 90 degrees and lightly sweep the loop 1 or 2 times through the inoculated area. And then streak to the next quadrant without overlapping the previous streaks, causing a dilution. Next is to flame and cool again. Next is to flame the loop again like this. Then turn the plate to another 90 degrees and lightly sweep into the third quadrant of the plate. Then flame and cool the loop once again, and then finally streak the last quadrant remaining. Next is we are ready for the incubation. It is to allow your colonies to grow for 24 hours, placing the plate in the incubator set for a temperature of 37 degrees Celsius, then wait for the result for at least 24 hours. Our main objective is to choose bacteria from one of these pure colonies and create a pure culture using a straight plate method. The fourth quadrant is usually where you will find the isolated colonies like this picture. Because this is because with each successive quadrant, the inoculum was further diluted. So that was all. Thank you very much, and I hope you learned everything what I have just said, like what I've learned it from my teacher. Thank you.